Imagine if you felt completely disembodied. Imagine you couldn't tell that you were in pain, that you were experiencing a comforting hug, or the wind on your face.
It is fundamental to how we understand, we grasp the world.
Touch and pain give us the sense of our own body.
Even if we close our eyes, our body knows where it is. And that's part of the somatosensory system.
It guides how we move and how we perceive and interact with the world. Perhaps more than any other sense, it gives us our sense of self.
These senses are critical for our survival and emotional well-being. So just from a basic science perspective, it was interesting.
For more than 150 years, neuroscientists have tried to understand how the nervous system enables us to sense touch and pain.
The somatosensory system is often underappreciated, but it's really fundamental, not just for our survival, but also to how we understand ourselves as humans.
For more than a decade, the Brain Prize has celebrated groundbreaking discoveries in neuroscience. Today, it is the world's largest award in the field.
The Brain Prize celebrates the people behind breakthroughs that transform neuroscience. I'm very inspired by scientists who dedicate themselves to uncovering fundamental truths about things we take for granted. This year, we honor 2 scientists who revealed what gives us our sense of self.
Which scientist's outstanding work has been recognized by the world's largest award for neuroscience? Now we reveal the winners of the Brain Prize 2026. From an exceptional field of nominations from all areas of neuroscience and countries around the world, the selection committee has chosen 2 scientists to receive this year's Brain Prize. They're being recognized for pioneering research that transformed a whole field by revealing how the nervous system detects and processes touch and pain. The Brain Prize 2026 goes to Professor Patrik Ernfors and Professor David Ginty.
Winning the Brain Prize to me is remarkable. I love my work. I love what I do. I love being a scientist. I love being a professor. So it's an honor to be recognized for really my life's work.
I feel very proud being the recipient of the Brain Prize. It's the most important recognition there is in neuroscience. I was very surprised and, of course, deeply honored.
Andreas Meyer-Lindenberg, you're the chair of the Brain Prize Selection Committee. What did the selection committee find so compelling about this year's award winners?
Together, they have really changed the textbooks. When I was studying, the thing that was in the textbooks was that there is a single neuron that connects these sensors in the skin with parts of the brain. And this year's laureates have made us understand that there is a broad spectrum of sensory neurons and that they start to interact very early on. So they've made a groundbreaking advance in understanding one of the basic building blocks of how we sense the world. They have overturned orthodoxy, and what we find in the textbook now is what they have found.
The status of the field when I entered was that we knew that there were different kinds of sensory neurons, but we didn't really know how many they were and how different they were.
My goal was really to understand how the peripheral nervous system develops. And I realized about 10 years into this work that we were studying the development of a system, but we really didn't understand fully the cells and circuits that underlie the mature somatosensory system. So that's what I really became interested in.
There was a huge technological development at that particular time that actually suddenly made it possible to create a taxonomy of sensory neurons.
Having genetic access to them allows us to study their properties across the body, their functions, how they connect centrally to convey their signals up to the brain? What are the pathways that they take from the entry of these signals into the spinal cord, to the brainstem, to the midbrain, to the cortex? These are all these questions are made more tractable, if you will. We can answer them more in greater detail.
David Ginty is a professor of neurobiology at Harvard University, studying the biology of touch. His work focuses on low-threshold mechanoreceptors, the neurons that are involved in the perception of light touch.
To understand how the sense of touch really works, we have to understand how all of these low-threshold mechanoreceptors work collaboratively. There's really not a mechanical stimulus other than high-frequency vibration that relies solely on one type of mechanosensory neurons. So when you stroke your hand across the skin, we think that there are at least 5 and maybe 6 different low-threshold mechanosensory neurons that are activated. We're still trying to figure some of these out, but now that we can study them in great detail, we know more about properties of individual cell types. Today, we think that there are at least 20 different sensory neurons, spinal sensory neurons, that make up the somatosensory system. Now we have great, deep molecular biological insight into all these sensory neurons. It's just opened up a whole range of beautiful directions for us to study to understand how the somatosensory system works.
Patrik Ernfors is a professor of tissue biology at Karolinska Institutet, studying the cells and circuits that drive pain. His research aims to understand how pain arises and how this knowledge can guide the search for better treatments for chronic pain.
Chronic pain affects society massively. It changes your entire world. Your daily life is just centered around the pain. It affects your mental condition, it affects your sleep. So what we did was using single-cell RNA technologies, being able actually to obtain the full molecular insights into those cells and developing animal models where we can actually manipulate the function only on those cells. Once you classify them, you actually know every single gene expressed in that cell type. So now you can actually go in and start understanding, just looking at the genes being expressed, you can make a lot of different predictions of the functions of the different neurons. And based on those predictions, you can design experiments that now actually provides evidence for that predicted function, which I hope is gonna, and we're working towards, is gonna drive the field forward so that we can find new treatments for chronic pain.
We're also interested in what happens when the somatosensory system doesn't work properly. Why are children with autism spectrum disorders often overreactive to light touch? With that information comes the possibility of identifying druggable targets in these sensory neurons that will allow us to tone down their activity to treat some of these sensory overreactivity disorders. That's really what's exciting to me, is that we're asking questions that have been around for, in some cases, hundreds of years, and getting answers to them.
Together, Ernfors and Ginty have really redefined how we think about somatosensation. Their work has been extraordinarily elegant and incisive on the level of just how neuroscience is done, and yet so impactful in the way this addresses very current, timely and urgent question that we encounter in the clinic. They have really provided highly important tools and made these tools broadly available to the scientific community, therefore bringing the field forward.
At Linköping University in Sweden, Professor Håkan Olesen and his team are among the researchers using advanced methods to uncover how touch and pain signals are processed in the human nervous system, tracing their path from skin to brain in both health and disease. Why is the work of the winners so fundamental to the field?
I think it's fair to say that their methods have revolutionized the studies of the somatosensory system. They have looked all the way from molecules up to behavior in a very systematic way. They demonstrated the diversity of all the different sensory neurons. We have specialized neurons for detecting fine hair movements, pulling of hairs, painful pulling of hairs, and caress-like slow touch movements over the skin. They have unfolded or demonstrated the logic of the somatosensory system.
And how has their work contributed to your own research into the sense of touch and pain on humans?
They have added new tools to the field, very new important tools for other researchers to use. They have made life much more easy for me because we can see what they have found in their animal models and see if that applies to humans. So they have inspired me to ask questions based on their findings in our research on humans.
And what does it mean for the future of research and discovery in this whole field?
What I hope is that we now can develop drugs that can treat conditions like chronic pain. And there are also diseases like polyneuropathies, the degeneration of the peripheral nerves following diabetes or chemotherapy against cancer. So might even be important for how we view somatosensory symptoms in different psychiatric diseases. They have given us the basis, the map, the framework for start developing new treatment strategies. That's how I see it.
The work of the winners of the Brain Prize 2026 has provided researchers around the world with a new roadmap for understanding the sense of touch and pain, one that is already reshaping the field and inspiring the next generation of scientists.
I have big hopes for the future. I think it can really give inspiration to a new generation also that can continue driving this research forward.
This prize to me represents really the work of the people that I associate with. It's my students, so it makes it extra meaningful. It's really a prize for them.
Dear David, dear Patrik, on behalf of the selection committee and the Lundbeck Foundation, my warmest congratulations on winning the Brain Prize 2026.